Nearly there now. Question 8. So this one had to do with the nucleic base pairs that you have. If you want, pause the video to take a minute to reread the question. One of the key parts of that question was for you to rethink which ones were actually complementary and what does that actually then mean when you're looking at numbers. So key bits to remember, and that has to go into your answer. So identify the types of bases. Types of bases. So which ones were purines, which ones were pyrimidines? That's your first out of the 3 marks, okay? So just saying that we have got C and T. These ones here are the pyrimidines, and A and G are the purines. How do you remember what's what? The purines, I remember the letter U is the one with the double ring. The pyrimidines are the ones where pyramids of Giza, where you've got 3 pyramids. So you've got 3 of them. You've got thymine, cytosine, you've got uracil as well. So therefore I remember that the 3 of them were the pyrimidines. There are lots and lots of other memory aids. I'm pretty sure you've got some already, okay? And you've always, out of a base pairing, you've got a purine and a pyrimidine with each other, because obviously A and G being purines, they have got the double, double has got a U in it and purine's got a U in it. So they have got a double carbon, so double nucleic acid ring, so double base, and these ones are the single rings that are in there. So if you know that you have to have A and T always pair and G and C always pair, what does that actually tell you here? It tells you that if you have, why is that percentage here? So the C and T added together over A and G added together. Why is the ratio of pyrimidines the same? The ratio of pyrimidines to purines, why is that always at 1? Because you always have them bonding complementary. So that is the key idea that has to go there, that they're complementary. The last part of the question here is, why do we have these ratios here different? So why do we have how many A's we have, which automatically pays with T's, is different to how many C's there are compared to those? That literally is that there's a different sequence of whatever is in maize has a different gene or a different number of genes there present compared to the human liver cell. So let's look at that on the mark scheme as such. If you see it as a mark scheme, so C and T are the pyrimidines and A and G are the purines. Because DNA is double-stranded and you always have the complementary pairing, you therefore need to have the same number or percentage of purine as pyrimidines. That needs to match up because they're complementary. And the last one, there's no necessity in the number of adenines being in any way related to, let's say, the number of cytosines, because they're different nucleotides, and therefore you could have a different number of each making up that particular sequence, whether it's the human liver or whether that's the maize. It might be AAAATT and no C's and G's in sight anywhere, randomly, because of whatever gene there is around and available. The second question here, suggest why the ratio that you're supposed to have for the C and T to A and G, so this one here, why is that not exactly 1? It should be. Why is it not precisely 1? It should be 1. This is only down to experimental error. So something might have been contaminated, something might have been lost in transfer. There is no other explanation. Simply no other explanation apart from loss in transfer or experimental procedural error. So experimental error, loss of material in the process. This one here, I am sure that methylation is something that you will have looked at already. So the idea that part of a gene can be silenced is the wrong expression, but a gene can be, by being methylated, it will no longer be transcribed as frequently. Now, methylation is taking place on the level of the actual nucleotide, so on the level of the nuclear base. So instead of a normal cytosine, you'd have a methylated cytosine, which has got a couple of changes. Irrelevant what the changes are, but it's got a couple of changes, which is methylated, just calls it methylated. The information that it gives you Exposure to some chemicals can cause those modifications. Genes with a high proportion of these modified bases may not be transcribed. May, not won't be transcribed, but may not be transcribed. So first question here, explain why this modification to the DNA would not result in a change to the structure of the protein. It's still cytosine. It is still cytosine, so it will be read as a cytosine. It will, in the process of transcription, be simply copied and copied over and still be used as cytosine, and therefore translated and later on transcribed the same way. So you'd have still the same amino acids picked to make that protein. Very, very last question. Actually, let me just go to the mark scheme here. For the second from last one, change of cytosine does not alter the DNA base sequence, so no effect on the primary structure because you haven't altered the DNA sequence. And very last question here. What change would this have on the phenotype, so the actual appearance of a person? If a gene, if it's methylated, may not be transcribed, that means you're probably making far less of the protein that it's supposed to stand for. So let's say you've got a protein like the color in your iris of your eye, or the pigment in your skin, or the curly protein that makes your hair curly, or whatever it also makes your hair straight. If that gene is not transcribed as much, we're not going to have as much messenger RNA, and therefore not as much much protein produced on the actual ribosome, which means that the appearance will be changed. This is an epigenetic thing. I am sure that you have learned enough about epigenetics that actually, as much as we think DNA is something that is fixed and stable and doesn't get changed, the environment switches genes on and switches genes off, not only internal, but it's external factors as well that switch genes off and switch genes on if required. So let's look at the mark scheme as the last one for this. It's an epigenetic change because the DNA changes the ability of the gene to be transcribed, maybe or maybe not transcribed more, probably less. So therefore the protein would not be produced or produced as much. Therefore the appearance, or whatever you have got the gene for, it might be a gene for a particular digestive enzyme. If that doesn't get produced, then that enzyme isn't produced, obviously, as a protein being an enzyme. And therefore whatever substances need to be changed by the enzyme no longer work. So the structural role or the functional role that enzyme would play, or the appearance role, would no longer be there if that protein doesn't get produced.